Thank you everyone for joining us today. As mentioned, we will be talking about the new Local Run Manager version 3 that comes with MiSeq Control Software version 4. As the agenda for today's talk, I'm going to give a quick introduction to MiSeq Control Software, or MCS version 4, and then talk about Local Run Manager, or LRM version 3, give an overview of that, and then do a deeper dive into the features of Local Run Manager version 3, talking about accessing and using Local Run Manager, setting up runs and analyzing data, how that interacts with the MiSeq Control Software to actually start the run, and then how you view your analysis results after that. We also have some resources at the end of the slides, and then we'll have time for questions as well. So let's start with a quick introduction to MiSeq Control Software version 4. So this is the new version of MiSeq Control Software that comes bundled with the Windows 10 update. As you may be aware, we've been updating our instruments from Windows 7 to Windows 10 due to the Windows 7 end of life from Microsoft. And part of that, in terms of the Windows 10 update, means we have a new version of MiSeq Control Software. We'll talk about how to navigate that a little bit later on in the talk, but most of the functionality is still pretty much the same, but you'll notice it does look a little bit different. As part of the new version, we still have the touchscreen graphical user interface. We have a drop-down menu where many of the functions that were previously on the launch screen have been moved to. And we have video clips that demonstrate how to load flow cells and reagents, and different ways to start the run. And we'll show all of those as we get to that part of the talk. So first, let's give a broad overview of Local Run Manager and version 3 in particular. So if you've used Local Run Manager before in any of our other instruments that have it on the instrument, then many of these features are going to be the same or familiar to you. So Local Run Manager provides an on-instrument integrated solution for user management, run creation, to set up your runs, and to analyze the data from your runs, and also to view your results. Local Run Manager starts out as a framework that has common parts such as analysis engines, dashboards, and user management. And within that framework, you then install specific analysis modules. So if you're coming from MiSeq Reporter and MiSeq Control Software version 2, this is a slightly different design philosophy. With MiSeq Reporter, everything was released all at once in a software suite. With Local Run Manager, you have the framework and you only install the particular analysis workflows you want to use. After you set up the run, that run information is sent to the instrument control software when you select that run configuration. The run is performed, sequencing data is generated by RTA. That data is sent back to Local Run Manager to perform your secondary analysis. Now, just to let you know, there are no changes to RTA or the primary data analysis in the Windows 10 update or MiSeq Control Software 4. So if you have older analysis pipelines that you're already using, there's no change to that data coming off of the instrument, and you can still use those secondary analysis pipelines. Now, as mentioned, this is a modular architecture, so this has the benefit of allowing us to update specific components, usually specific analysis modules, with updates or bug fixes without needing to update or revalidate the entire software suite. And important to note that on the MiSeqs with the new control software, Local Run Manager version 3 replaces MiSeq Reporter. So MiSeq Reporter is not installed with the MCS 4 update and cannot be co-installed with Local Run Manager 3. This is a replacement of the older MiSeq Reporter software. As mentioned, there are other versions of Local Run Manager, and depending on the instrument type you have or which control software you have installed, you may have different versions of the software. So this is just to break down what each one of those are. The initial release of Local Run Manager was part of the MiSeq Control Software and version 1. So MiSeq Control Software had version 1 of Local Run Manager as well. That was part of the Windows 7 version of that instrument. The DX instruments, MiSeq DX and NextSeq DX, have their own particular versions of Local Run Manager on the DX drive. And then version 2 is what's on most of the other instruments and currently available off instrument. So that is part of the MiSeq if you updated to MiSeq Control Software version 3. Comes shipped with the iSeq 100. If you've updated an older MiSeq to version 2 or you purchase a new MiSeq now, it will come with Local Run Manager version 2. Same for the NextSeq. Instruments that have been updated to Windows 10 and the latest control software also have Local Run Manager version 2. Version 3 is a new version that comes with this version of the MiSeq control software. There is also an off-instrument version planned that will be released soon, but as of today is not yet available. We also are releasing modules specific to version 3 over a staggered release schedule. As of today, we have 3 modules available: Generate FastQ, Library QC, and Resequencing, and we will be releasing more over time. We'll have a support bulletin out soon on our support bulletin site that gives a timeline for when each module is going to be released. Before you update your MiSeqs, we do ask that you communicate with your field representatives and fill out a pre-install checklist and let them know which particular workflows that you normally use on your MiSeq. If the particular workflow as a module is not available yet, but will be available shortly, you may want to delay your update, but that's a conversation we recommend having with your FAS. I mentioned different versions of the modules. The modules and workflows are specific to each framework. So you cannot take the version 1 or version 2 modules and install them in version 3. Cannot install the DX modules in the RUO frameworks, and vice versa. In terms of the off-instrument versions, only one version can be installed on the same PC. You cannot, say, co-install version 2 and version 3 of the off-instrument versions. Now, with all of these different framework versions and different module versions, we know it can be a little bit confusing to figure out which module you need to download for your version. So we have released a tool that is available on our support site, the Local Run Manager Module Selector tool. And there's a couple of different ways to get to it, and we've included links here. What this does is allows you to start from the particular instrument you're using or the off-instrument version of the software you're using, and then go down to control software version, framework version, and particular module you want. And this will give you the direct links to the module for that version that you need. And this is where we recommend going to make sure that you get the correct module for the Local Run Manager framework version that you're using. Doing so will also give you links to the software guides, the framework guides, and any other product files that may be applicable, such as sample sheet templates. Installing modules is similar to our other instruments that use Local Run Manager. You would download the module, move it to see Illumina, and unzip the file. And then from there, run the MSI installer from that unzipped folder. The first thing you'll see is Local Run Manager credentials, and there's a couple ways to install this depending on your account management state. We will talk about account management in a little while, but if you have that turned on, and that means that you have to log into Local Run Manager with the username and password, that will apply to your module installation as well. So if you have that turned on, provide the administrative credentials for Local Run Manager. So these are the Local Run Manager credentials for your admin account on that, and not your Windows login credentials. So something to keep in mind. If you have account management turned off, which means you would just normally connect to Local Run Manager and load the dashboard with no login, then you would also not provide any account information here. You would leave these blank as shown in the screenshot and just hit next. Let's talk about these features specific to Local Run Manager version 3 and why we have a version 3 instead of iterating version 2 for the MiSeq. So there are new versions of Local Run Manager, new features in Local Run Manager version 3. The first thing you'll notice is the decoupling of the library prep kits and index kits. In previous versions, you'll have a single drop-down for selecting the kit that contains both the library information and the index information. If you've used other of our software, such as Illumina Experiment Manager or Instrument Run Setup in BaseSpace for the NextSeq 1K, 2K, you'll notice that there's two selections. That's because these have been decoupled, where you select the library prep kit that has information such as supported run sizes and adapter sequences. And then from there, you can select compatible index kits. This reflects how you can order different combinations of prep kits and index kits depending on your need. So that's reflected in the software itself. We've also added a password recovery option. So if you've ever been locked out of your local Run Manager account because you forgot your password or put in the incorrect password too many times, we have now the option to put in recovery questions. So this is the same kind of idea that you may have, say, on a website for your bank or any other kind of site that uses a login where you would put in, what is your first pet's name, etc. It's the same kind of idea here. Set recovery questions that are personal to you, and you can recover access to your account. If you're still locked out of that, though, we do have the option to have you call into technical support, and we can help you unlock the account that way. There are also various networking and security updates, such as better support for HTTPS certificates, better support for connecting an instrument to an off-domain server, and other updates that are outlined in the release notes. Those are already posted on our website. Recommend you read through them to see all the changes. Let's talk about now how you actually access and use Local Run Manager. Local Run Manager is accessed through a web browser on the instrument. You're pointing the browser to the same instrument. So if you're familiar with using MiSeq Reporter or previous versions of Local Run Manager, the default address is just localhost. This means you are connecting the instrument to itself. So while this uses a web server, it does not connect to anything off-site. So you do not need an active internet connection in order to use Local Run Manager. You can also access the on-instrument interface through a web browser on the same network. So say if your instrument is in one room and you're in an office down the hallway, you can access the interface remotely to set up runs or look at results, either through the instrument name or using the IP address. The interface for this is optimized for Chrome or Chromium. It also works well with Firefox, but Internet Explorer is not recommended. You will usually see rendering problems with that. So as mentioned, you can connect to this remotely. If you want to find out what the instrument name is, you can just go to the dashboard and you'll see it here at the top. You can also get the IP address of the instrument just from the Windows command line. Depending on your networking setup, one may be better to use than the other, such as if your instrument has a dynamically assigned IP address. Networking questions are also part of that pre-upgrade checklist that you would send to your FAS. They can help you determine which system or which method may be better for you. We mentioned account management when it comes to installing modules, so I want to talk about it here again before we get into setting up runs, because how you set this does impact how you set up runs and also how you start runs. So in the Tools drop-down menu from your dashboard, you'll have account management that you can turn on or off. And you can set different user accounts as either admin or users, and they have different privileges and different functions that they can do. If you have account management turned on, then you will need to log into Local Run Manager before you can access the dashboard. If you have it turned off, then when you put in the address or local host, you will automatically load the dashboard. This also affects how you start runs. If you have account management turned on, when you go to start a run in the control software, you will need to log in with your Local Run Manager credentials to access ready runs. If you have that turned off, you'll just see all your ready runs available for selection. And when we go through how to select runs later in the presentation, you'll see there that we're not logging into Local Run Manager. We have account management turned off in that case, as that shows us all the runs that are available without needing to log in. So let's go through the different ways you can set up runs and then how that leads to analyzing your data. This is a broad overview of the workflow. So optionally, again, if you have account management turned on, you would log into Local Run Manager. Once at your dashboard, you would select Create Run, enter in the details and settings, add in your sample information, and save your run. When you get to the control software, you'll see that there are 3 ways to start your run. If you're familiar with MiSeq Control Software version 3, then this will look very familiar to you. So you have Local Run Manager mode, which allows you to select runs you set up in Local Run Manager that we'll be going over shortly. You have sample sheet mode, which is most similar to MiSeq Control Software version 2, where you select a sample sheet to configure and start your run. And then you have manual mode, where you do not use a sample sheet, you just put in cycle numbers that only generates raw data. We do recommend local run manager mode, as this will make sure that your run setup has no errors or anything that will cause problems with analysis. Now, one very important thing, and if you remember really only one thing from this presentation, it's this, is that sample sheets generated from Illumina Experiment Manager are not compatible with this version of the control software. There are formatting differences between IEM sample sheets and how Local Run Manager version 3 formats sample sheets or inputs information, so you cannot use IEM sample sheets with Local Run Manager version 3. We know this may be a large change in workflow from previous versions. We're trying to sort of make this transition as smooth as possible, first by letting you know this. So if you try to import an IEM sample sheet, you will get error messages, both in Local Run Manager or in sample sheet mode in the control software. Because of this, we do recommend setting up runs in LRM mode. If that's not convenient or the most efficient workflow for you, you can use a Local Run Manager formatted sample sheet. And on the support site, with each module, we do have template sample sheets that you can use. Download that template, fill it in. If you have a system where you programmatically generate your own sample sheets, you can use those Local Run Manager version sample sheets as models to update your format. Also, when you download a module, that module installer comes bundled with a template sample sheet that you can also fill in and upload or use in sample sheet mode, or look to see the formatting differences. So just keep this in mind as we go through the rest of the presentation that we talk about sample sheets. They are Local Run Manager formatted sample sheets and not IEM sample sheets. So we do have two ways to set up a run in Local Run Manager, either entering in run details manually or importing a sample sheet, again, a Local Run Manager sample sheet and not an IEM sample sheet. And we'll go through both methods. First, the manual method. So here you would connect to Local Run Manager and load up your dashboard, and you select Create Run, and you will see a drop-down list of the workflows that are installed. You will select the analysis workflow that you want to use. Now, if you are looking for a particular workflow and you don't see it in that drop-down, that means that that module is not installed. Go to our Local Run Manager support site, use the module selector tool, find the module you need, install that module, reload the page, and then you'll have it available for selection. Once you select the module, in this case we're using Generate FastQ, you'll see the screen options to either import a sample sheet or enter in the run details manually. When you do this, anything with the red asterisk is required information, so you would enter in a run name, and run names must be unique on the instrument. Descriptions are optional. Those are mainly for your information. You would select the library prep kit, followed by the index kit. Change anything you need to about read cycles or read types. And if you're using custom primers, you can set that here. One thing to keep in mind is that each library prep kit does have default read lengths. So if you start with one library prep kit, change the read lengths, and then change to a different kit, then note that those read lengths will reset to their default values, and you may need to change them again. The index lengths will depend on the kit you select and will be detected from what you enter in the sample ID area in a little bit later. So you also have module-specific settings. These do vary by analysis workflow. In Generate FastQ, you generally just have adapter trimming you can turn on or off. But in something like DNA Amplicon, you would have the option to change your aligner or change your variant collar or settings about that variant collar. The different options available are described in the workflow guide for each module. After that, you would enter in your sample information, put in your sample IDs, select your indexes, and then you can add in any other rows or samples that you need to. We talked about how library prep kits and index kits are decoupled in this version of the software. So the library prep kit option is where you select common information, read types in terms of single or paired end, index schemes, read lengths, and what the compatible index sets are. So after you select the library prep kit, that will then populate the index kit, giving you the compatible index selections for that library prep kit. After you set that up, you can enter and run details manually. Again, anything with an asterisk is required. You would include a unique sample ID, and Local Run Manager will live validate whatever you put in here. So if you put in any illegal characters, such as spaces or plus symbols or slashes or something like that, you'll get an error message and not be allowed to save the run until you correct it. And this does make sure that you do not have any downstream errors from your initial setup. You would select your index information, and those index combinations need to be unique. If you repeat the index combinations, you'll again get an error message and need to correct that before you can continue. Different modules or different workflows may require other information, such as a targeted panel may require a manifest and a genome to be selected. Once that information is selected, you would click on Save Run, and then your run would be ready in the dashboard. You also have the option to export the sample sheet for further use. So let's go through an example here. We're going to click Create Run, select the Generate FASTQ module. We're going to enter in our run name, select our library prep kit. We can make any changes to read types or read lengths, as well as note if we're using any custom primers. Select the index kit we're going to use. We can turn adapter trimming on or off. And then we're going to enter in our sample information. Select our indexes. And then we see the Save Run becomes ready at the bottom. We can also change to show the sequence instead of the name if you need to confirm that you've selected the correct indexes. Once you click Save Run, then you go back to the dashboard and it shows that you're ready. That was how you would set this up manually. Note that you can also add multiple rows, and if you have, say, a spreadsheet with your sample and index information ready, you can copy and paste. If you have a sample sheet, again, a Local Run Manager formatted sample sheet, you can use that to import your information. We mentioned a few slides ago, but I'll remind you again, IEM sample sheets are not compatible with MCS version 4 and Local Run Manager version 3. You can create a template through LRM. You saw that export sample sheet option in the previous demonstration, but we do also have templates available on our support site. Using the sample sheet option, you can import a compatible sample sheet with your information, make sure the run name is unique, and then click on Save Run. So here's an example of that. Going to choose Create Run, again, Generate FASTQ for our example. Going to import a sample sheet. Select our example run here. And then we see our selections are filled in already with our library prep kit, read lengths, and all of our different samples. We just click on Save Run, and then our run is ready for sequencing. We talked about selecting library prep kits or sample sheets. As introduced in the previous version of Local Run Manager, you do have a couple of different options for custom entries. So you're not limited to just the standard Illumina kits. One of the options is to select custom from the library prep kit dropdown, and this then allows for free text entry of indexes. So here you can use any index sequence you want, limited to AT, GC, and ENDs. So in Local Run Manager, ENDs are used as wildcards, and this custom option is the only place where you can enter in ENDs. Index links must be consistent between samples. What this means is that every sample needs to have consistent lengths between them. So they all need to be listed as 8x8 or 6x6, or they can be non-same in terms of you can have 6 and 8 as long as they're all consistent between the samples. Now, we usually don't recommend mixing index types, and we do have some good bulletins that explain why. But if you do have different mixed index lengths in your samples, you can NPAT in Local Run Manager, and those will be used as wildcards. Note that if you're uploading to BaseSpace, BaseSpace uses different software for demultiplexing, and does not recognize ends as wildcards, your demultiplexing will not work the same if you use ends in base space, and you would need to use separate sample sheets there. So these are just considerations to keep in mind. Now note that index 2 should always be in the forward orientation. This is on the MiSeq, so that's not really something you have to worry about, since I5 is always read in the forward orientation. But as mentioned, there will be an off-instrument version of Local Run Manager 3, and that is compatible with data from our other benchtop instruments. So that would be the iSeq, the MiniSeq, and the NextSeq. It's not compatible with our high throughput instruments, such as the HiSeqs or the NovaSeqs or the NextSeq 2000. But if you are using the off-instrument version, always enter in your index 2 in the forward orientation or the MiSeq orientation. The software will recognize the data type depending on the run files and automatically reverse complement the I5 sequence if necessary. Besides selecting custom from the drop-down menu, we do have the option to add your own custom kits if you're using third-party or homebrew kits repeatedly. But there are 2 files you need to download, make changes to, and upload back to the software now. There is a library prep kit and the index kit, and you can download templates from the interface or download the files that are already used by installed kits if you just want to make changes or have something similar to current kits. You would upload the library prep kit first, and then upload your index kit that references back to the library prep kit that you're going to use for compatibility. And that index kit is what contains your index sequences, and that determines your index links. And once you set up the run, you do have the option to change a few things before you start. So there is the edit option where you can change how your run is set up in terms of your samples or index selections. There is an import option that is similar to version 2, and that allows you to analyze existing data, and we'll talk about that a little bit later. If you do need to make any changes, just click continue to save it after that. The examples we showed before were just generate FASTQ. Some workflows do require manifests for our targeted panels, such as DNA amplicon, RNA amplicon, or DNA enrichment. And there's 2 ways of loading those manifests. If you run the same panel repeatedly, you may benefit from going to the modules and manifest section from the tools dropdown and adding the manifest to that particular module. When you do this route, that allows the manifest to be selected from your run setup each time you go to that module. The other method is the ad hoc method. This is when you're setting up a run. Once you're in your data section, you can use the import manifest option to import the manifest for this particular run. This will make sure that the manifest shows up for selection from that drop-down menu, but it will not be available for future runs unless you import it again, or as shown in the previous slide, import it into the module. The instrument will have pre-installed genomes, and those are instrument dependent, but you can use genomes that you either download from iGenomes or custom genomes. And again, if you're familiar with Local Run Manager version 2, it's the same kind of way you do it. So within the Local Run Manager settings, you have a genome repository path. By default, that is C Illumina genomes, but you can change that path to another drive, which is useful if you are using an off-instrument installation and need room on another drive for that data. If you're downloading the information from iGenomes, you'll have the folder structure already correct. If you're installing a custom genome as a FASTA file, you need to make sure that the files are in this folder structure of species, agency, version, whole genome FASTA, and then genome.fa. That's also described in the software guide and is again the same as version 2 if you're familiar with that. So we've talked about how you actually set up runs. Let's look at how you start your run once you're ready to sequence. So when you start up MiSeq Control Software version 4, you'll note that it looks a little different from version 3 or version 2, but does look very similar to some of our other Windows 10 instruments, such as the NovaSeq or the MiniSeq that's been updated to version 2. You'll see this blue interface with a sequence button and a wash button. The run options that you normally would see from the previous versions are just relocated to this drop-down menu. So if you click on that or use the touch screen, that's where you'll see the options for doing system checks, to manage your files, reboot, etc. In terms of starting the run, you would just start from the sequence button. Once you select sequence, you'll see these three options that we mentioned before: local run manager, sample sheet, or manual. Local Run Manager allows you to select a run that you set up in Local Run Manager through one of those methods that we showed you before. Let's take a look at what this looks like. So here we select Local Run Manager. We have the option to set up base space for either run monitoring or full data upload. Here we're not using that, but we just click on Next, select that run that we set up before. We have the option to confirm that these are the correct samples before we start. Here we're going to go on to Next. You see the video to load the flow cells, load the PR2 on waste, load the reagent cartridge. You have the option to preview your run, make any changes you need to, change your output folder. If you realize these are the wrong lengths, you can change them here. Otherwise, you'll do your pre-run checks and then start the run. The second option is sample sheet mode. Now, a reminder, again, IEM sample sheets are not compatible with MCS4, even in sample sheet mode. You do need to use the Local Run Manager formatted sample sheet. So here you would select sample sheet. Again, you have the option to upload to base space. In this mode, you also have the option to disable local secondary analysis. Here you would select the sample sheet that you want to use. By default, these are located in MiSeq Control Software sample sheets. Local Run Manager will then take a few seconds to fully validate the sample sheet. So this is being validated by the Local Run Manager module that the sample sheet calls. And if you have any error messages or any problems, those will be shown here. But if it's correct, you just go on through the same procedure of loading the flow cell, reagents, and starting the run. The third option is manual mode. Manual mode does not allow for full run data upload. There is no sample sheet, so you do not have any sample information, but you can still use base space for run monitoring. So your metrics will be uploaded to base space, but not the run data, and you will not be able to demultiplex in the cloud in manual mode. In manual mode, you enter in your read types, single read or paired end, enter in your read links for read 1, index 2, and read 2. And note that you do need to enter in your index cycles manually in this case because those are not being read from the sample sheet. If you forget to put in cycle numbers and the run continues, that data may not be recoverable because you may not have captured that information. You also have the option for loading custom primers here. Note that in this current release of the software, you do not have the option to select custom recipes. That may be introduced in a future update, but stay tuned to see what happens with that. So now you've performed your run, you've set up your secondary analysis, everything is completed. How do you actually view your results? So when you go back to the Local Run Manager dashboard, you can select on the run. Analysis will begin once sequencing and primary analysis are complete. Note that the instrument can only perform sequencing or analysis at once, cannot do one while the other is in progress, and preference will always be given to sequencing. So if you have an analysis in progress and you start a new run, you'll be asked if you want to stop the first analysis. You can select yes and start your run. At that point, you would need to go back and restart your analysis once the run is complete, and that will restart from the beginning. It will not pick up where it left off. Analysis time will vary based on the workflow you've selected, the complexity, and the overall amount of data. So here's an example of a run that we've selected from the dashboard. You'll see these three tabs. The first is the run overview that gives general information about the run. You can see such information as total clusters, clusters passing filter, and other high-level information. Note that this is not an SAV replacement. If you want to see detailed metrics, you can still use Sequencing Analysis Viewer, but this does not give you all the same charts as SAV does. Also pointing out the output run folder line here, this is the path to where your actual files are located. So in terms of your FASTQ files or BAM files or VCF files, So if you want to get to those, the easiest way to do that is click on that first button next to the path, which is a copy button. That will copy the path. You can open a Windows Explorer window, paste that in, and that'll take you directly to your output. If you need to change the output path, say from a local drive to a network drive or vice versa, you can change it here and then requeue analysis, and it will regenerate the files and copy them to the new output location. The second tab is the sequencing information tab. This is, again, kind of a high-level overview, gives you common information about the flow cells and reagents that you used. The samples and results tab is where you're going to get an overview of your results. And what you see here is going to differ depending on the workflow that you chose. And these are most similar to the PDF reports you would get from BaseBase with your overall results. So you can see an example here of RNA amplicon where it gives you percent-aligned reads, gives you an overview of coverage, and so on. Depending on the particular workflow, you can expect specific outputs, and we do recommend looking at the workflow guide to see what you should expect. So overall analysis starts from your raw data, like previous versions of Local Run Manager and MiSeq Reporter. It does not start from FASTQ files, but will generate them from your BCL files each time you start analysis. Depending on the workflow, you can then have other steps. For this example, the DNA amplicon, you will align to your reference and identify your variants. Output files will then include demultiplexing information, FASTQ files, BAM files, VCF files, and so on. Your output files will vary depending on module, and again, new modules will be released to the support site, and we recommend using the module selector tool to find the one that you need. You do have the option to edit a run after sequencing, and this can be for a few reasons. You may need to change your sample information, say if you forgot a sample or put in the wrong sample or chose the wrong indexes. You can go back, use the requeue option, edit the setup, make whatever changes you need, and then requeue analysis. You do have the option to reanalyze existing data in a new workflow or new module. This is the process you would follow if you need to change the workflow that you're using. Say that you initially set up the run as FASTQ only, and now you want to perform enrichment analysis in this example. You would set up the run with that information, go to the Actions menu, select Import, provide the run folder path, and in an output folder path, and import the run. If you're familiar with version 2 of Local Run Manager, this is again the same process. Once you provide a valid input and output path and import the run, analysis will start automatically with that new workflow. If you're monitoring the run in Local Run Manager, there are several possible states, and these are color-coded. So if you just see ready or no particular color here, that just means that the run is set up and ready for further input. Anything in the blue status means something is underway. Sequencing is running, complete, or analysis is running. Red means something has gone wrong. So either the sequencing itself was unsuccessful or there was some problem with the analysis. Green means everything is complete and is hopefully what you see at the end of your run. There are some resources we want to point you towards to get more information. So here's a link to our Local Run Manager support site. And from there, you can get the software guides and workflow guides and installers for each of the modules. And again, we recommend using the module selector tool, which you can get to from this link, to make sure that you're looking at and downloading the correct version for your framework. We also have a new training video released from our training site that goes into further detail about how to get started with Local Run Manager version 3, how to set up runs. It's about 20 minutes, and it's a good review of just everything we've talked about today, and more information about how to start your runs and look at your results. We went over a lot of topics today, so wanted to give you this final slide as kind of a digest for the important points that we talked about today. So with the MiSeq Control Software version 4 update, Local Run Manager 3 replaces MiSeq Reporter. We still will have MiSeq Reporter and Local Run Manager version 2 available for off-instrument use, but on-instrument, that is replaced by Local Run Manager version 3. Analysis workflows are installed separately as modules. Again, when you're getting ready to do the update, we have a pre-install checklist to go over with your FAS. Let them know which workflows you use to make sure that those are installed before you start your first run. IEM sample sheets are not compatible with this version of the software. You can download sample sheets from our support site, create runs in Local Run Manager, or download templates from Local Run Manager itself. You can also set up runs remotely. So this is useful if you want to use Local Run Manager mode but are not in front of the machine. As long as you're on the same network and the instrument allows for network access, you can just use the instrument name or IP address, set up your run, and then that will be ready to select once you're in front of the instrument. And with that, we'd have time for any questions.